Oogenesis begins with the female germ cell, the oogonium, see Fig 3.1. Oogony.1 rapidly divide by mitosis to produce primary oocysts. While oogonia are present in adult ovaries of some species, amphibians. Mammalian oogonia exist only before birth. Once a female mammal is born, her primary oocytes are arrested at prophase of first meiosis, and cannot be ovulated until she is sexually mature. A typical primary oocyte is characterized by a large germinal IDC, nucleus, with a germinal pot, nucleolus, in the ooplasm, cytoplasm, and is bounded by the oolgma, plasma membrane. Primary oocytes of many species enter a growth period where the oocytes stockpile many organelles and other cellular components including ribosomes and yolk. The primary oocyte divides by first meiosis to produce a large Neurulation, and a small first polar body which houses the extra set of maternal chromosomes in a small quantity of ooplasm bounded by plasma membrane, Fig. 4.1. The secondary oocyte undergoes meiosis II to produce a large mature ovum with a female pronucleus, haploid nucleus, and the small second polar body, which houses the remaining extra maternal chromosomes. Female gametes of different animals can be ovulated, released from the ovary, at any stage of meiosis. When ovulated during meiosis, the female gamete is called a primary or secondary oocyte. When ovulated after meiosis is complete, it is called an ovum. Oogenesis is seasonal in many species, echinoderms, fish, amphibians, birds, and under hormonal control. Some mammals, rodents, dogs, pigs, horses, sheep, exhibit an estrous cycle, which in rats, lasts 5 days and continually repeats itself. Thus, an adult female rat ovulates several oocytes every 5 days. Sheep have a seasonal estrous cycle, which occurs in the fall. Primates exhibit a menstrual cycle, which lasts -28 days in humans. The estrous and menstrual cycles have two major functions: they cause release of a mature female gamete, and prepare the endometrium, uterine lining, for possible pregnancy. In primates, 1. Oocyte is typically ovulated, released from the ovary, per menstrual cycle, but in other organisms such as the roundworm, Ascaris, up to 200,000 oocytes are released daily. Ovarian anatomy. Oogenesis occurs in the ornery, female gonad, which is located within the chylome, body cavity or abdomen. There is a diversity of ovarian structure, size, and shape. For example, most mammalian ovaries are paired, small oval organs of equal size and function, Fig 4.2. In contrast, only the left ovary functions in birds, whereas in sharks and certain bats, only the right ovary functions. During development, the two ovaries fuse to form one large gonad in lampreys and certa and fishes, Osteichthyes, whereas one ovary fails to develop in hagfishes, medaka, and guppies, leaving these species with only one functioning ovary. At the other extreme, many echinoderms have 5 or more elongated ovaries. The two Ascaris ovaries are thread-like, being long and thin. During the breeding season, teleost and amphibian ovaries take up a large volume of the female's body. The ovarian surface is smooth in some species, but more often during the breeding season it is nodular, bulging with developing oocytes. Reptilian and avian ovaries resemble a bunch of grapes. When not actively cycling, the surface of mammalian ovaries is smooth, but most become nodular when active, Figs 4.3, 4.4. Some mammalian ovaries have a narrow end called the hilus. The mesovarium, mesentery, holds the ovaries in place within the body cavity by attaching the hilus to the round ligament and Herode ligament, which is a layer of peritoneum, mesentery, that attaches the uterus to the dorsal and ventral body wall, Fig 4.2. Closely associated with the mammalian ovary is the oviduct, fallopian tube. The ovulated oocyte is picked up by the phymitria, open end of oviduct, travels through the infundibulum, distal region of oviduct, and then the ampulla, middle, and isthmus, proximal area of oviduct, before entering the uterus, Fig 4.3. In some vertebrates, snakes, birds, there is only one functioning oviduct, instead of two, and its orifice, the ostium, is located relatively far from the ovary in amphibians. Ov.Arian histile. OGY. He ovary is composed of two main cell types, female reproductive cells, oogonia to ova, and supporting tissue, epithelium, connective tissue, vascular system, follicle cells. Vertebrate oocytes exist in the ovarian cortex, periphery, surrounded by follicle cell, granuloma and Thuca cells, derived from connective tissue, Figs 4.4, 4.5. In many species, follicle cells provide nutritional support, produce hormones, and aid in yolk buildup in the oocyte. An oocyte and its associated follicle cells are called a follicle. Some follicles grow and mature, eventually releasing their mature oocytes during ovulation, whereas other follicles are rudimentary, never co-release their oocytes. Several types of follicles can be observed in the ovary that reflect the maturity of the enclosed oocyte. As the primary oocyte matures, follicle cells proliferate mitotically to form multiple layers that surround it, increasing follicle size. Most of the primary oocytes in the ovary possess a large germinal vesicle. As the oocyte grows, the germinal vesicle is located off-center. In mammals, the most mature follicles, Graafian follicle, are easy to identify, since they develop a large antrum, follicular cavity, full of fluid, liquor folliculi. In chickens, the most mature follicles contain extremely large oocytes, greater than 30 mm, full of yolk. Once they have undergone a period of growth, oocytes can be released from the ovary at any stage of meiosis. Avian and mammalian oocytes arrest at meiotic prophase, until hormonal stimulation resumes meiosis. The oocyte is ovulated as a secondary oocyte, which arrests at metaphase of meiosis 2 until fertilization. In contrast, starfish oocytes are ovulated at the primary oocyte stage, whereas sea urchins shed ova, meiosis is complete, during spawning. Oogenesis in model organisms. The appearance of ovaries greatly varies. A common theme is that oogenesis begins with oogonia that mature into oocytes/ova that are released from the ovary. Vertebrate ovaries can be classified into two types: the compact type, which is characteristic of teleosts, reptiles, birds, and mammals, and a saccular type found in amphibians. The compact vertebrate ovary has two major zones: the outer cellular cortex, where oogenesis occurs, and the inner medulla, made of fibrous loose connective tissue that is populated with fibroblasts, nerves, and large blood and lymph vessels, Figs 4.4, 4.5. In mammals, a connective tissue layer, the tunica albuginea, surrounds the ovarian cortex. The germinal epithelium covers the entire vertebrate ovary and is continuous with the mesovarium. In the saccular type of ovary, the cortex and germinal epithelium are as described for the compact ovary, but the medulla area is hollow, forming an ovarian cavity or lymph space. Selected organisms that are excellent models to understand oogenesis are described in this chapter. Echinoderm. Echinoderm ovaries are lobular, Fig 4.6. The starfish ovary is located in the chylome of each arm. A thin visceral peritoneum surrounds each ovary, Figs 4.7, 4.8. Oogenesis begins with oogonia located in the basal region of the germinal layer at the periphery of the ovary. The oogonia mature into primary oocytes that are released from the germinal layer and stored in the ovarian lumen until they are spawned through gonadopores. A difference between sea urchin and starfish ovaries is that sea urchin ova are released from the germinal epithelium and stored in the ovarian lumen at the pronuclear stage when meiosis is complete, rather than at meiotic prophase. Lancelet, Amphioxus, Branchiostoma. The paired sac-like ovaries become packed with primary oocytes during the breeding season, Figs 4.9, 4.10. Eventually the ovaries burst, releasing their mature primary oocytes into the atrium. Water flowing posteriorly in. The atrium carries the primary oocytes to the atriopory where they are released from the female during spawning, Fig 4:11. Frog. The paired ovaries are multilobular and saccular, raking up a large volume in the ventral abdomen during the annual breeding season. The myxovarium, double-layered extension of the peritoneum, attaches the ovaries to the body wall and covers each ovary as rhe thucextrina. The ovarian oocytes are surrounded by inner follicle cell LS and outer theca cells which form the theca incrina, Figs 4:12 to 4:16. Oocytes are arrested in meiotic prophase for a long period, up to 3 years in Rana papiens, as the oocyte grows. During this time, the oocyte and its germinal vesicle stockpile materials needed for later embryogenesis. The diplotein germinal vesicle is characterized by lamp brush chromosomes active in transcription, and multiple nucleoli involved in ribosome synthesis, Fig 4:15. After the first 1.5 years, the Rana oocyte begins to accumulate yolk, made by the liver, and stores it in the ooplasm as yolk platelets during a process called Vitellogenesis. Oocytes of some species, Rana, Xenopus, are pigmented at one end of the oocyte cortex, and pigment masses are visible within the ovarian lumen. By the time they are mature, primary oocytes increase in size about 1,000-fold. Upon appropriate hormonal cues, the primary oocytes resume meiosis and then arrest at metaphase of second meiosis. Soon they are ovulated into the chylome where ciliary movements of the chylome wall move the secondary oocytes to the ostium of the oviduct. Several jelly layers are then placed around the oocytes during their passage through the oviduct. The jelly layers function to protect the oocyte and developing embryo from mechanical injury and infection, and serve to aid in fertilization. Secondary oocytes are stored in the ovisac, uterus, for about 1 day until their release during oviposition. Rana papiens release 2,000 to 3,000 oocytes that are 1.75 mm in diameter each during oviposition, whereas up to 20,000 secondary oocytes are released from Rana catesbiana. After spawning, the ovary contains only oogonia and supporting tissue, and thus the ovary is significantly smaller. Bird. In the adult hen, Gallus, only the left ovary and oviduct function. The right ovary and oviduct are rudimentary. The ovary is located near the kidney. Oocytes exist in follicles composed of inner granulosa cells, stratum granulosum, and outer theca cells that form the theca interna and theca externa, Figs 4:17 to 4.23. The granulosa cells secrete an inner perivitelin layer, IPVL, which is a fibrous glycoprotein layer surrounding the ooloma. Both the oocyte and its nucleus increase in size during the growth phase as the oocyte stockpiles materials including organelles and yolk. The germinal vesicle has lamp brush chromosomes active in transcription, Fig 4.20. In mature primary chicken oocytes, the germinal vesicle is found within the 3 mm wide germinal disc, yolk-free area of cytoplasm. About 6 hours before ovulation, the germinal vesicle breaks down. The first polar body is extruded about 1 hour before ovulation. When ovulated, the secondary oocyte is arrested at metaphase of second meiosis. A typical ovary of the mature hen of the chicken has several ovarian follicles based on their color and diameter, 1,000 or more small white, 1 mm, 20 large white, 5 mm, 10 small yellow, and 5 to 7 large yellow, up to 36 mm. Mammal. The tunica albuginea encloses the cortex of the mammalian ovary, which surrounds the deeper medulla. Unlike the model species discussed thus far, the mammalian follicle is characterized not only by growth of the oocyte, but also by an increase in the number of follicle cells as the oocyte matures, Figs 4.24 to 4.34. The oocyte is surrounded by a stratified layer of granulosa cells called the stratum granulosum, which becomes surrounded by an outer layer of theca cells called the theca folliculi, Fig 4.5. As the follicle grows, the theca layer differentiates into a vascularized inner theca increna, and an outer fibrous theca stemma. Both the granulosa cells and theca interna secrete estrogen. The germinal vesicle of the oocyte is large and has lamp brush chromosomes active in transcription, Fig 4.34. During each estrous cycle or menstrual cycle, a cohort of follicles is stimulated to grow, but only one or a few of these become mature follicles, the rest become aderctic and degenerate, Figs 4.35 to -4.39. As follicles mature, the oocyte becomes surrounded by the ona PCLLM ida, a 2 to 25 m extracellular coat composed of 3 or more glycoproteins, ZPL2, ZP3, which function in spermoocyte interactions during fertilization. The zona pellucida is surrounded by granulosa cells called cumulus cells, corona radiata of the cumulus oophorus, which remain attached to the zona pellucida during ovulation, Fig 4.40. The freshly ovulated oocyte and surrounding cumulus cells are called the cumulus oocyte complex. In most mammals at the time of ovulation, the oocyte has progressed through first meiosis and is arrested at metaphase of meiosis II, and thus is a secondary oocyte with a well-developed zona pellucida, Fig 4.41. After ovulation, follicular cells, from the ovulated follicle, that remain in the ovary become a corpus luteum, Latin, yellow body, which secretes progesterone and estrogen to maintain the endometrium in case pregnancy occurs, Figs 4.4 to 4.5, 4.42 to 4.43. The corpus luteum enlarges further, up to 5 cm in humans, if pregnancy occurs. If not, the cells of the corpus luteum die and become scar tissue called a corpus albicans, Latin, white body, Fig 4.44. The following is a summary of the major structures found in the cortex of the mammalian ovary. Primordial, immature, follicle, primary oocyte surrounded by one layer of flattened follicular, epithelial, cells called granulosa cells, no antrum, Figs 4.24 to 4.25. Primary, growing, follicle, primary oocyte surrounded by one or more layers of cuboidal granulosa cells, no aneurysm, Figs 4.26 to 4.28. Secondary follicle, primary oocyte surrounded by many granulosa cell layers that are surrounded by layers of theca cells derived from the ovarian seroma, antrum is forming, Figs 4.29 to 4.31. C. Renophion, prion 1 later or mature, follicle, secondary oocyte surrounded by corona radiata and many cumulus cells, follicle wall is multi-layered with granulosa and theca cells, large antrum is present, Figs 4.32 to 4.34, 4.36 to 4.37. Atretic follicle, degenerating follicle with an antrum, numerous dying cells with dark, pycnotic nuclei present, Fig 4.35. Corpuluteum, formed from follicular cells remaining in the ovary after ovulation, becomes a major endocrine organ. The large granulosa, luteal and thecal luteal cells stain pale since they have a large quantity of smooth endoplasmic reticulum in the cytoplasm that synthesizes mostly progesterone, Figs 4.42 to 4.43. Corpus albicans, remains of the no longer functioning corpus luteum, Fig 4.44. Stroma cells, connective tissue cells found between follicles and corpora lutea, Figs 4.24 to 4.26, 4.28. 4.30 to 4.33, 4.37. Tunica albuginea, thin, dense irregular connective tissue sheath surrounding the ovarian cortex, Figs 4.24, 4.27, 4.31, 4.32, 4.35 to 4.39. Germinal epithelium, simple cuboidal epithelium covering the tunica albuginea. It is the external surface of the ovary, Figs 4.24, 4.27, 4.31, 4.32. 4.35. Fertilization is the defining process of sexual reproduction when a new individual with a unique genetic constitution is produced. It is the union of the female gamete, oocyte, ovum, and the male gamete, spermatozoon, to form a single-cell embryo, the zygote. Fusion of the haploid gametes restores the chromosome number to the diploid complement of chromosomes, and activates the female gamete so that development is initiated. This classical definition of fertilization has been modified, depending on the species studied. 1. The events from spermoocyte contact leading up to and including the point of spermoocyte fusion. Spermoocyte contact to first mitosis, and 3. Spermoocyte contact to the point when the maternal and paternal chromosomes are first together within a common nucleus, which in mammals occurs at the two-cell stage. The second definition is used in this atlas. Essential events of metazoan fertilization include gamete activarion, biparental heredity, and production of a viable zygote. Fertilization is cl-chronal in some species such as echinoderms and most frogs. Oocytes, ova, are shed by the female into an aqueous environment and are subsequently fertilized by sperm released from a nearby male. Other species, sharks, birds, mammals, have internal fertilization. Sperm are deposited in or near the female reproductive tract and fertilization occurs within the female. There is a species difference in timing as to when the embryos are released from the female reproductive tract. In oviparous animals, example some sharks, turtles, birds, embryos are released soon after fertilization so that they complete development outside the mother's body, nourished by the yolk. Alternatively, in ovoviviparum animals, example teleost-live-bearing guppy, the embryos develop in the oviduct nourished by the yolk until they hatch in the uterus and are then born. In viviparous animals, eutherian mammals, the embryos develop in the uterus, nourished by the mother until birth. Animals with internal fertilization usually produce fewer embryos than those with external fertilization. The source of energy for the developing embryo is reflected in the quantity of yolk in the oocyte, Table 5.1. Oocytes with little yolk are isolchital. In some organisms with isolecithal oocytes, echinoderms, the embryo develops relatively early into a self-feeding larva. Viviparous organisms tend to have isolecithal oocytes, since the mother will nourish the embryos until birth. Amphibian oocytes are mxolchital, having a moderate amount of yolk, which will support the embryo until it develops into a self-feeding tadpole. A large amount of yolk is needed in telelecithal oocytes of some oviparous animals, birds, and in centrolecithal oocytes of insects, since only the yolk will nourish the embryo until hatching. Like a planet with north and south poles, an oocyte has two poles, an animal pole which contains the nuclear chromosomes and associated cytoplasm, and a vegetal pole which is the site on the oocyte opposite the animal pole, Fig 5.1. An imaginary line that connects the animal and vegetal poles is referred to as the animal-vegetal axis. An imaginary line across the oocyte's equator reveals an animal hemisphere containing the maternal chromosomes with the animal pole at its center, and the vegetal hemi/fur that contains the vegetal pole. In oocytes of many species, fish, frogs, birds, the vegetal hemisphere has more yolk in the cytoplasm than the animal hemisphere. Entry of the fertilizing sperm in the larger mesolecithal and telelecithal oocytes is usually restricted to the animal hemisphere. Since isolecithal oocytes are relatively small, up to 2 pm, and have little yolk, they can be fertilized usually at any point on the oocyte surface. Oocytes of different species are released from the ovary, shed, spawned, ovulated, and fertilized at different stages of meiosis, Fig 5.2. For example, certain mollusks, surf clam, are fertilized at meiotic prophase, germinal vesicle stage, starfish at metaphase of meiosis I, most mammals at metaphase of meiosis II, and sea urchins at the pronuclear stage when meiosis is complete. The term oocyte is used when the unfertilized female gamete is undergoing meiosis, and ovum, ova, plural, applies to the unfertilized female gamete in which meiosis is complete. The term egg is more appropriately applied to cuisine, however, it is commonly used to refer to both the oocyte and ovum. It will be used here to refer to the domestic chicken egg, eggshell containing membranes, albumin, yolk, and oocyte. Oocytes are typically surrounded by one or more external coats which the sperm must penetrate before fertilization. A vitellin layer, vitellin envelope, made of glycoproteins surrounds the oolgma, plasma membrane of oocyte, of many species. Outside the vitellin layer of echinoderms is a thick extracellular coat called the jelly layer, jelly coat, which serves to attract and/or activate sperm, Fig 5.3. Frogs have 3 to 6 jelly layers which the oviduct deposits around the oocyte after ovulation. The vitellin layer of mammalian oocytes is the zona pellucida, which is composed of ZPL, ZP2, and ZP3 glycoproteins, see Fig 4.1. Spermatozoa are well equipped to reach the oocyte and penetrate through these extracellular coats, Fig 5.4. Fertilization involves several steps over a period of time, thus there is no single moment of fertilization. To prepare for fertilization, the sperm undergoes a series of activation events which allow it to become competent to fertilize the female gamete. These changes include sperm motility. Sperm become vigorously motile or change their pattern of motility, capacitation, a process that causes biochemical changes in the sperm and occurs only in mammals upon incubation in fluids of the female reproductive tract, and the actosome reaction. The acrosome reaction exposes receptors on the sperm that bind to the oocyte, and releases proteolytic enzymes that digest through the oocytes. Extracellular coat, Figs 5.5. 5.6. Once sperm hay acrosome reacted, they are competent to fuse with the oolema. Without these changes, a sperm cannot fertilize an oocyte. The intricate details of these events are beyond the scope of this atlas, and will be described only briefly. Sperm-oocyte interaction triggers a series of events in the ooplasm, cytoplasm of oocyte, that activate the oocyte and initiate development. First, the oolema depolarizes, becomes positively charged, or hyperpolarizes, becomes more negatively charged, depending on the species. In sea urchins, membrane depolarization prevents poly-PCRMY, more than one sperm fertilizing the ovum. Next, the oocyte releases calcium ions from the endoplasmic reticulum. This triggers the cortical granule reaction, which releases proteolytic enzymes and mucopolysaccharides outside the oocyte, Fig 5.7. The cortical granule reaction results in alteration of the vitellin layer, which lifts off the oocyte surface. This forms the fertilization envelope, which is a physical barrier that prevents polyspermy. After sperm-oocyte fusion, the zygote undergoes a series of additional morphological and biochemical changes to initiate development. Female pronuclear development. Meiotic maturation is completed. The maternal chromosomes remaining in the ooplasm after extrusion of the second polar body decondense, unravel, into a female pronucleus. In some species, sea urchins, this step occurs before fertilization. Sperm nuclear decondensation. The incorporated nucleus of the fertilizing sperm has highly condensed chromosomes. The sperm nucleus loses its nuclear envelope, undergoes chromatin remodeling, and decondenses. When the paternal chromatin has acquired a new nuclear envelope, it is called a male pronucleus. Formation of the sperm aster. Sperm centrioles of many species are surrounded by the centrosome, which nucleates microtubules to form the sperm aster. Male and female pronuclei migrate along the sperm aster to the center of the zygote, Figs 5.8, 5.9. Pronuclear fusion. Male and female pronuclei fuse to form a single zygote nucleus. In Ascaris and mammals, pronuclei do not fuse but lie juxtaposed, and the maternal and paternal chromosomes first intermingle after the pronuclear envelopes dissolve during mitotic prophase. Synthesis of DNA, RNA, and/or proteins. These components are needed for subsequent embryogenesis. The timing of synthesis varies among species. First, mitosis produces two daughter cells that proliferate and differentiate to form the multicellular adult. Fertilization in model organisms. Several model organisms are used in this chapter to demonstrate aspects of fertilization. Although there is diversity in the process of fertilization when comparing species, there are also similarities. There is a size difference between the gametes, with the sperm being significantly smaller than the oocyte/ovum. Also in the model organisms shown here, the male gamete is motile, and migrates to and fuses with the oocyte/ovum. Nematode fertilization. He-female reproductive system of the roundworm. Ascaris is tubular and Y-shaped, Figs 5.10 to 5.12. Each tip of the arm of the Y is a narrow ovary which gradually widens into the oviduct and ends in a dilated uterus. The base of the Y is the muscular vagina which opens to the exterior via the vulva, genital pore, located mid-ventrally 1/3 the distance from the mouth. Highly productive ovaries can produce up to 200,000 oocytes daily. In transverse section, the oviduct is wheel-shaped, containing triangular-shaped, isolecithal oocytes with the germinal vesicle in the broad end toward the oviduct wall. When released into the uterus, the oocytes become spherical with the germinal vesicle centrally placed. Roundworm sperm are atypical in that they lack a flagellum and use amoeboid motion for motile, Fig 5.13. After copulation, sperm migrate from the vagina to the uterus where they fertilize the primary oocytes at meiotic prophase. They are readily observed between oocytes in a section of the uterus, Fig 5.14. The sperm delivers its condensed, haploid nucleus and chemrioles to the ooplasm during fertilization, Fig 5.15. The sperm nucleus is relatively large, dark, and conical, and migrates to the oocyte center with the help of the sperm aster. When the maternal chromosomes complete meiosis, the female pronucleus migrates to the oocyte center. Pronuclei do not fuse prior to first mitosis, but lie together. The first time female and male chromosomes are located within a common nucleus is at the two-cell stage. The oocyte cortex changes after fertilization, producing a fertilization envelope and a thick chorion. The oocyte shrinks within the chorion shell, taking up only about 70% of its original volume. Most roundworms are oviparous. Echinoderm fertilization. Sea urchin. J. Hen sea urchins spawn, their gametes are released from their dorsal side into the surrounding sea water through 5 or more gonadoporcs, also called gonopores or genital pores, Fig 5.16. Ova are fertilized in sea water when a nearby male releases his sperm. The ova are isolecithal and 74 m in diameter in the sea urchin, Arbatia punctulata. The ovum contains a haploid, decondensed nucleus called a female pronucleus, which is about 10 m in diameter, see Figs 5.3, 5.8, 5.9, 5.17. The ooplasm is full of extra organelles and stored macromolecules which are required to carry the embryo to a feeding larva stage. Some ova have yellow, orange, or red pigment in the ooplasm. Two extracellular coats surround the ovum, a thin vitellin layer adjacent to the ooloma, and a thicker, clear jelly layer composed of mucopolysaccharides outside the vitellin layer. The sperm is significantly smaller than the oocyte, Figs 5.17, 5.18. The head of the Arbatia sperm is typically 3.25 m long and 0.75 m wide. It houses the sperm nucleus, a chromosomal vesicle, centrioles, and a small amount of cytoplasm containing globular, unpolymerized, actin. Within the acrosomal vesicle are proteases and bindin, see Fig 5.6. The Arbatia sperm tail is 45 m long and 0.2 m wide. Microtubules and dot-associated proteins provide the structure of the flagellum. A motor protein, dynein uses ATP to provide the force for sperm propulsion. All the ATP the sperm needs is produced by the single mitochondrion located in the sperm's midpiece, which is 0.75 m long and 2 m wide. To conserve energy, sperm are immobile in the testis. Upon their release into seawater, sperm become motile. They can swim for an hour or more depending on the environmental conditions, but eventually lose their energy. Therefore males usually release sperm when sea urchin ova are nearby. When a sea urchin begins spawning, others nearby sense this, and they too begin to spawn, thus ensuring a new population. To successfully fertilize an ovum, a sea urchin sperm must swim to and recognize only a sea urchin ovum and not that of any other species, which may also be present in the seawater. Sea urchin sperm are chemoattracted to the ovum's jelly layer and acrosome react in it, see Figs 5.5, 5.6. Interaction of bindin with the ovum receptor is species specific, allowing a sperm to fertilize only the same species. Ovum. Sperm binding to the oocyte receptor induces a change in electrical conductance of the ovum. Within 5 seconds, the tip of the acrosomal process fuses to the ooloma. Within 1 to 2 minutes post-insemination, MINPI, the cortical granule reaction occurs. This can be verified by the dramatic lifting of the fertilization envelope, Fig 5.19. At about 3 to 7 minutes pi, the point of sperm entry is marked by the fertilization RNNC, which is a temporary protrusion of the ooloma containing a microfilament core that aids in engulfing components of the fertilizing sperm, Fig 5.20. It is resorbed by 10 minutes pi. Sperm nuclear decondensation in Arbacia is completed by 20 minutes pi when the male and female pronuclei fuse to form a one-zygote nudu. By 60 to 120 minutes pi, the zygote enters first mitosis. Monospermy occurs when a single sperm fertilizes an ovum. To ensure monospermy, the ovum has N-O mechanisms to prevent polyspermy. 1. Early or Photo block to polyspermy, membrane depolarization of the ovum, and 2. Late or slow block to polypermy, cortical granule reaction, which leads to the formation of the fertilization envelope. Polyspermy in most organisms is disastrous because there are too many chromosomes and centrosomes delivered to the ovum, Figs 5.21-5.23. Starfish. Often spawning in warm shallow tidal pools at low tide, starfish, Asterias 4bc, have isolecithal oocytes about 110 m in diameter that are surrounded by a jelly layer. The oocytes contain a pale yellow pigment and are shed at meiotic prophase, germinal vesicle stage. When they come in contact with seawater, oocytes immediately resume meiosis, dissolving the germinal vesicle within 10 minutes, forming the first polar body by 1.5 hours, and the second by 2 hours, Fig 5.24. Sperm penetrate the oocytes at any stage of meiosis, but the best period for successful fertilization is after germinal vesicle breakdown, but before extrusion of the first polar body, Fig 5.24c. Sperm acrosome react in the jelly layer, forming a relatively long, thin acrosomal process which fuses with the ooloma, see Fig 5.5. In response, the ooloma forms a fertilization cone at the site of fusion, which aids in engulfing the sperm components. The oocyte also undergoes the cortical granule reaction, forming a fertilization envelope that begins to spread outward from the site of sperm entry. A sperm aster forms and moves the male and female pronuclei to the oocyte center, allowing them to fuse prior to first mitosis. Teleost fertilization. Any teleost fish, zebrafish, medaka, spawn just after the onset of daylight. Oocytes of these species are fertilized externally with milt, sperm. The telelecithal oocytes are 1 to 1.5 mm in diameter, and are arrested at metaphase 2 of meiosis until fertilization. The oocytes are decussal, sediment to the bottom, rather than pelagic, free-floating. While zebrafish let their oocytes immediately sediment, see chorionic, filament attach a cluster of 0 to 30 medaka oospheres to the female's vent for several hours before they sediment. Both species produce optically clear oocytes covered by a transparent chorion. Since the chorion is impenetrable by sperm, sperm can only enter the chorion at the micropilk, which is a small opening of the chorion above the animal pole, Fig 5.25. In zebrafish, the unfertilized oocyte appears homogeneous in that the yolk is epically distributed throughout the ooplasm, Fig 5.25a. Within minutes after fertilization, contractions of the oocyte cause yolk-free cytoplasm to segregate at the animal pole and form the B-elsta disc, a yolk-free zone in the oocyte, Fig 5.25b. The unfertilized medaka oocyte contains several oil droplets. AMPH. Avian fertil. IZA. TION. Only grown oocytes of the leopard frog, Ranopapiens are 1.75 mm in diameter. The oocyte cortex of the animal hemisphere is covered with dark melanin pigment granules, which protect the chromosomes from damaging rays of UV light. The vegetal hemisphere is creamy white since it contains yolk and lacks melanin, Fig 5.26. Sections through the unfertilized oocyte show the pigmented cortex of the animal hemisphere, and the grainy appearance of the yolky ooplasm, Fig 5.27. The vitellin layer surrounds the oocyte, and 3 jelly layers surround the vitellin layer, Fig 5.28. Unfertilized oocytes are arrested at metaphase 2 when they are ovulated and fertilized. During fertilization, a sperm penetrates the jelly layers before fusing with the ooloma in the oocyte's animal hemisphere. After gamete interaction, the ooloma depolarizes, serving as a fast block to polyspermy. The slow block to polyspermy involves cortical granule exocytosis, which triggers formation of the fertilization envelope. Spermoocyte fusion releases meiotic arrest and the second polar body forms as meiotic maturation is completed. Under the influence of gravity, the fertilized oocyte rotates so that the less dense, pigmented animal hemisphere is uppermost. This event is called the rotation of activation at fertilization. About 1 to 2 hours after fertilization, the entire oocyte cortex rotates 30 degrees toward the site of sperm entry, leaving behind an unpigmented area of cortical cytoplasm called the gray crescent, Figs 5.26, 5.28, 5.29. This cortical rotation is distinct from the rotation of activation at fertilization. Fertilization sets up the axis of the embryo in that the gray crescent marks the dorsal side of the embryo, and the site of sperm entry is the ventral side. Sections through the fertilized oocyte reveal that as the sperm journeys through the ooplasm, it drags with it melanin pigment granules which mark the path of migration of the decondensing sperm nucleus, Figs 5.29, 5.30. The sperm aster forms allowing the sperm nucleus to migrate toward the developing female pronucleus. The pronuclei fust, prior to first cleavage. In some anurans, Puerto Rican tree frog, ecoquy, and many urodels, salamanders. Newts, the oocyte is normally polyspermically fertilized, however, only one sperm contributes its genome to the embryo and the rest degenerate. Avian fertilization. Heterolecithal avian oocyte is covered with an inner perivitellin layer, IPVL, that forms a fibrous supportive meshwork made of glycoproteins, Fig 5.31. For successful fertilization, sperm penetrate the IPVL using acrosomal enzymes to digest small areas of it. Freshly ovulated oospheres are arrested at metaphase 2 until fertilization, which occurs near or within the infundibulum of the oviduct. The oocyte chromosomes are located in the germinal disc or blastodisc, yolk-free cytoplasm. Sperm fertilize the oocyte at the germinal disc, within 15 minutes after ovulation. While some species are normally polyspermically fertilized, all but one sperm degenerates. Only the surviving sperm contributes its genome to the embryo. After sperm entry, meiosis resumes and is completed. Pronuclei form by 3 hours after ovulation. Mammalian fertilization. Mammalian sperm must first capacitate before they can acrosome react. Capacitation occurs in the female reproductive tract. Oocytes are fertilized in the ampulla, distal portion, of the oviduct. Upon binding to the zona pellucida via ZP3, capacitated sperm acrosome react releasing proteolytic enzymes, hyaluronidase and acrosin, from the acrosomal vesicle that digest a hole through the zona pellucida. The acrosome reacted spermatozoon next binds ZP2, and then fuses with the oolema by the sperm's equatorial segment, on its side, rather than with the tip of the acrosomal process as in sea urchins. Spermoocyte fusion causes a calcium release in the oosphere that triggers the cortical granule reaction. Proteolytic enzymes from the cortical granules biochemically change the zona pellucida glycoproteins in a process called the zona reaction. This prevents other sperm from binding the zona pellucida, thereby avoiding polyspermy. Oocytes of most mammals are fertilized at metaphase of second meiosis, and thus are secondary oocytes. They complete meiosis forming the second polar body soon after spermoocyte fusion. The sperm nucleus decondenses into a male pronucleus, which migrates to the oocyte center, Figs 5.32, 5.33. The maternal chromosomes that remain in the oocyte after polar body formation decondense to form a female pronucleus, which migrates to the oocyte center. The female and male pronuclei remain as distinct entities and do not fuse. During prophase the chromosomes condense and intermix on the first mitotic spindle. Cleavage occurs about 24 hours after fertilization. After mitosis, the male and female chromosomes are first found within a common nucleus at the 2-cell stage. During cleavage, the zygote, one-cell embryo, divides by mitosis into 2 daughter cells called blastomeres, Fig 6.1. Each blastomere soon divides into 2 more blastomers, resulting in a 4-cell embryo. The process repeats itself many times to produce a multicellular blastula embryo. Early embryonic cells usually enter mitosis faster than typical adult somatic cells, body cells that do not contribute to the germ line, because the embryonic cell cycle has shorter or non-existent G1 and G2 phases. The cytoskeleton plays a crucial role in cleavage. Microtubules, composed of tubulin, and associated proteins form the mitotic spindle. The mitotic spindle is needed for karyokin I, chromosome separation during mitotic anaphase, so that chromosomes are distributed equally to daughter cells. Microfilaments, composed of actin, form the contractile ring, which allows for cytokinesis, cell division, and helps create the Avage furrow, which is the physical constriction that divides the embryo into 2 daughter cells, Fig 6.1, 6.2. All embryos of different species cleave the same way or at the same rate. The amount and distribution of yolk, see Table 5.1, affects the cleavage pattern, Fig 6.3. For example, very little yolk is found in the isolecithal human oocyte, which divides after fertilization into 2 separate cells, holoblastic divage, of equal size, Fig 6.3. In contrast, the fertilized telelecithal chicken oocyte cleaves incompletely, meriblastic cleavage. Cleavage occurs through the blastodisc, but not through the yolky cytoplasm. Cleavage occurs faster in areas of the embryo with little yolk and is significantly slower in yolk-laden areas of cytoplasm. For example, in mesolecithal amphibian oocytes, the first cleavage furrow begins at the animal pole and ends at the vegetal pole. The furrow forms up to 50 times faster at the animal pole than at the yolk-laden vegetal pole. The pattern of cleavage is dictated not only by the amount of yolk in the oocyte, but also by the development and position of the mitotic spindle. Cleavage can be meridional, through the animal-vegetal axis, longitudinal, parallel to the animal-vegetal axis, but not through the poles, equatorial, through the cell's equator, which is perpendicular to the animal-vegetal axis, longitudinal, perpendicular to the animal-vegetal axis but not through the cell's equator, or at an angle oblique to the animal-vegetal axis, Fig. 6.4. Several variations can be found in the pattern of cleavage. Holoblastic cleavage, cleavage furrow completely separates both daughter cells, Fig. 6.3. Compare with meriblastic cleavage. Microblastic cleavage, cleavage is incomplete. Daughter cells remain connected, Fig. 6.3. After many cleavages, the embryo is a cluster of cells. Blastoderm, located on top of a mass of yolk. Compare with holoblastic cleavage. Equal cleavage, two equal-sized daughter cells result due to a central positioning of the mitotic spindle, Fig. 6.5. Compare with unequal cleavage. Unequal cleavage, one large and one small daughter cell are produced due to mitotic spindle placement at one side of the embryo, Fig. 6.sc, d. Compare with equal cleavage. Radial cleavage, cleavage plane is perpendicular or parallel to the previous one, Fig. 6.6. Compare with spiral cleavage. Spiral cleavage, cleavage plane is oblique as compared to the previous one, Fig. 6.6. Compare this with radial cleavage. Bilateral cleavage, first cleavage plane bisects zygote into right and left halves. Subsequent cleavages result in an embryo with two halves that are mirror images of each other, Fig. 6.7. Compare with radial and spiral cleavage. Rotational cleavage, first cleavage is meridional, during second cleavage one blastomere divides meridionally, and the other equatorially, Fig. 6.8. Compare with radial cleavage. Dicoidal cleavage, cleavage is restricted to the blastodisc of the telelecithal oocyte, Fig. 6.9. Compare with superficial cleavage, Fig. 6.9. Superficial cleavage, cleavage is restricted to a rim of cytoplasm in the centrolecithal oocyte, Fig. 6.9. Compare with discoidal cleavage. When the multicellular embryo is a solid ball of blastomeres, it is called a morula. Cleavage results in the formation of a multicellular blastula embryo, which is then ready to undergo gastrulation. The appearance of blastulae can differ, depending on the species examined, Fig. 6.10. 4. Example, blastulae of echinoderms resemble a hollow ball of a single layer of cells, codoblastula, whereas some cnidarian blastulae are a solid ball of cells, stereolastula. The space within a chylioblastula is the elastococcale. Cleavage in M-O D L
A O Organ
isms. Select number of model organisms are used in this chapter because they are well suited to study the variety of cleavage patterns. Their order of appearance in the chapter reflects the level of sophistication in the way their embryos divide. Echinoderm cleavage. Starfish produce isolecithal primary oocytes that undergo cleavage while surrounded by the fertilization envelope. Cleavage is radial and holoblastic, Fig. 6.11, 6.12. The first two cleavages are meridional, but at right angles to each other, and the third is equatorial. All cleavage divisions are equal and the blastomers are loosely arranged rather than in tiers of cells. The blastula is a hollow ball of cells that develop cilia, which allow the embryo to rotate within the fertilization envelope. The embryo hatches from the fertilization envelope at the blastula stage. Sea urchin. The small amount of yolk of the sea urchin ovum allows the cleavage furrow to penetrate through the cytoplasm relatively rapidly. While the first cell cycle lasts 1 to 2 hours, depending on the species, cytokinesis takes about 10 minutes or less, Table A5. After the first cell cycle, subsequent cleavages occur at -30-minute intervals. As shown in Fig. 6.13 to 6.15, cleavage is radial, holoblastic, and equal for the first three cleavages. First and second cleavages are meridional, and the third is equatorial. In the fourth cleavage, the four blastomers of the animal / top tier of the embryo divide meridionally and equally into 8 cells, while the 4 cells of the vegetal, bottom tier divide equatorially and unequally. This yields 4 micromegahertz, smallest cells, and 4 macromegahertz, largest cells, at the vegetal pole, and 8 megahertz, medium sized cells, at the animal pole. The micromers eventually become primary mesenchymal cells, which are the first to show gastrulation movements. The surface of the blastula becomes covered with cilia, which allow the embryo to rotate in the fertilization envelope prior to hatching. By 8 to 10 hours after fertilization, the blastula hatches out of the fertilization envelope and becomes free-swimming. Nemertean cleavage. Cleavage is spiral in nemerteans. In the milky ribbon worm, Cerebratulus, first cleavage is meridional and equal, Fig 6:16, 6.17, Table AL. Second cleavage occurs 20 minutes later, and is also meridional and equal, but orthogonal to the first cleavage plane. The third cleavage plane is unequal and oblique to the animal-vegetal axis. After several cleavages, a hollow blastula forms by 15 hours after fertilization. Molluscan cleavage. Cleavage in mollusks with isolecithal oocytes is pernell and typically holloblastic, Fig 6.18, 619, Table A.2. The first and second cleavages are meridional, pass through the animal pole at an oblique angle. The third cleavage is equatorial. Cleavages can be equal or unequal depending on the species. The second cleavage plane is oblique and either to right, dextra, or left, sinistral, of the first cleavage plane. In snails, the direction of cleavage influences the pattern of shell spirally ng in the adult. After about 6 cleavages, mollusk embryos form a blastula. In general, freshwater species form a blastocele, chylioblastula, whereas marine species do not, stereoblastula. Several members of the phylum Mollusca exhibit an unusual cleavage pattern unlike that discussed so far. For example, zygotes of the tusk shell mollusk, Dentalium, and marine mud snails, Nasarius, Ilionassa, form a transient, bulbous protrusion of vegetal cytoplasm immediately before first cleavage, Fig 6.20, Table A.2. The protrusion is called the polar lobe and is 1/3 the volume of the entire zygote. The polar lobe contains morphogenetic determinants. First cleavage is unequal and the polar lobe is associated with only one of the 2 blastomers. The other blastomere does not receive any of the morphogenetic determinants of the polar lobe. At this point the embryo looks as though it has 3 cells and is called the trefoil stage embryo. After first cleavage, the polar lobe is absorbed. Immediately before the second cleavage, a second polar lobe forms and is resorbed. Second cleavage is also asymmetrical, allowing the second polar lobe to remain associated with only one of the 4 resulting blastomers. In this way morphogenetic determinants are segregated into specific blastomers. Cephalochordate cleavage. Cleavage in the lancelet, Amphioxus, branchiostoma, is radial and holloblastic, Fig 6.21, 6.22, Table A6. First cleavage is meridional and equal. Depending on the species, second cleavage is either meridional, but at right angles to the first cleavage, resulting in 4 equal-sized blastomers, or slightly longitudinal, unequal, resulting in 4 cells with 2 being slightly smaller. Third cleavage is equatorial, or slightly latitudinal displaced toward the animal pole, and separates the blastomers into an upper and lower quartet of cells. Fourth cleavage is meridional producing an animal, top, tier and vegetal, bottom, tier of 8 cells each. Fifth cleavage is latitudinal and produces 4 octets of cells arranged one on top of the other, 2 tiers of animal octets, micromers, and 2 tiers of vegetal octets, macromeres. The sixth cleavage is meridional producing 4 crowns of 16 cells each. Cleavage is synchronous up to the 7th cleavage. The blastula stage begins after the 8th cleavage, 256 cell stage. The lancelet blastula is composed of a single layer of cells surrounding a large blastocele filled with a jelly-like substance, Fig 6.22. Teleost cleavage. Cleavage in teleosts, zebrafish and medaka, is meriblastic, discoidal, and equal, Fig 6.23, Table A.7. In zebrafish, the first 6 cleavages are synchronous and occur every 15 minutes at 28.5 degrees Celsius. First cleavage begins 40 minutes after fertilization. The first cleavage furrow is meridional and divides the blastodisc into 2 equal blastomers that re-and connected to the yolk, Fig 6.23b. The second through fifth cleavages are also meridional. Second cleavage occurs perpendicular to the first, so that a 2x2 array of blastomers is produced, Fig 6.23c. Third cleavage consists of 2 planes parallel to the first, and produces a x4 array of blastomers, Fig 6.23d. Fourth cleavage results in 2 cleavage planes parallel to the second and produces a 4x4 array of blastomers perched on top of the yolk, Fig 6.23e. Tracking dye studies have shown that the 12 outer cells called marginal cells are still connected to the yolk. In contrast, the 4 inner cells, non-marginal cells, are completely separate from the yolk. Fifth cleavage is meridional and results in 4 cleavage furrows that are parallel to the first. Fifth cleavage produces a 4x8 array of blastomers, Fig 6.23f. The collection of cells can be referred to as the halastod cream. In some fish, sea bass, trout, the non-marginal cells of the blastoderm divide latitudinally, while the marginal cells divide longitudinally during fifth cleavage. The sixth cleavage in zebrafish is latitudinal, passing through every cell of the blastoderm and produces 2 tiers of cells. All the blastomers of the top tier are completely separate from the yolk. The cells of the top tier, together with the marginal cells, are collectively called the enveloping layer, because they completely surround the inner, lower layer of deep cells called the deep cell layer. The seventh and subsequent cleavages exhibit asynchronous cell cycles and cleavage planes are no longer regularly patterned, Fig 6.24. The zebrafish blastula appears as a cap of cells on top of the yolk. Amphibian cleavage. Cleavage in amphibians is radial and holoblastic, Fig 6.25, 6.26, Table A.8. First cleavage in the African clawed frog, Xenopus laevis, is meridional and equal, and bisects the gray crescent. Second cleavage is also meridional and equal, but at right angles to the first cleavage plane. Second cleavage results in 4 cells, each with a pigmented animal hemisphere. Because of the greater amount of yolk in the embryo's vegetal half, the second cleavage furrow appears before the first cleavage furrow has completely penetrated through the vegetal hemisphere. Third cleavage is latitudinal, unequal, with the larger blastomers, macromercs, found in the vegetal half of the embryo and smaller cells, micromeres, at the animal half, Fig 6.26, 6.27. The fourth cleavage is meridional, and the fifth cleavage is equatorial. At this point the 32-cell embryo has 4 tiers of 8 cells each. The blastula has pigmented micromers at the animal hemisphere and pigment-free macromeres at the vegetal hemisphere. Multiple cell layers surround the blastocele that is displaced toward the animal pole. The floor of the blastocele is flat and bounded by macromeres, whereas its thin roof is arched and composed of micromers, Fig 6.27. The blastocele is filled with fluid. Avian cleavage. Avian cleavage is restricted to the blastodisc, germinal disc, a disc of yolk-free cytoplasm at the animal pole, which in chickens is 3 mm wide, Fig 6.28. Cleavage is microblastic and discoidal, Fig 6.29, Table A9. The first and second cleavages are meridional, and at right angles to each other in the center of the blastodisc. The second cleavage begins before the first is completed. The third and fourth cleavage planes are meridional and parallel to the first. Once the blastodisc is multicellular, it is called the blastod cream. At the 8 and 16 cell stages, the blastoderm is a single layer of cells which absorb fluid from the albumin and secrete it between themselves and the yolk, forming the subgerminal cavity which enlarges over time. Subsequent meridional, longitudinal, and equatorial, latitudinal, cleavages create a blastoderm that is up to 6 cells deep. Cells in the central region of the blastoderm become completely separated from the yolk by the subgerminal cavity. The deeper cells of the blastoderm eventually die, leaving a one-cell layer thick sheet of cells called the P-blast over the subgerminal cavity. The subgerminal cavity gives the central part of the embryo a lighter, translucent appearance, Fig 6.30. This central area is called the area pellucida, meaning clear area. The surrounding peripheral area of the blastoderm is still multilayered. It is connected to the underlying yolk and appears darker and more opaque than the area pellucida, hence it is called the area opaca. The junction between the area pellucida and area opaca is the marginal zone, marginal belt. The blastoderm next becomes a bilaminar blastula, in which cells that delaminate from the epiblast migrate into the subgerminal cavity, forming clusters of cells called polyinvagination islands, primary hypoblast, Fig 6.31, 6.32. Cells from the posterior of the marginal zone at Kohler's sickle migrate anteriorly into the primary hypoblast to form the secondary hypoblast. At the time the egg is laid, or soon after, the embryo is characterized by two distinct layers: 1, the P-blast, which is the surface layer of the area pellucida, after the hypoblast has formed, and 2, the hypoblast, which is the newly formed lower layer of large yolky cells that develops beneath the epiblast, Fig 6.33. The space between the epiblast and hypoblast is the blastocele. The subgerminal cavity is present beneath the hypoblast. As discussed in the next chapter, the epiblast gives rise to the embryo proper, whereas the hypoblast produces portions of the extraembryonic membranes. Cleavage stages occur in the oviduct, Table 6.1, while several accessory structures are deposited around the oocyte by the oviduct, Fig 6.28, 6.34, 6.35. The vitellin membrane is the transparent protein layer surrounding the egg yolk. It consists of two closely associated layers, an inner and an outer perivitellin layer. The ovary produces the inner perivitellin layer, IPVL, prior to ovulation, while the infundibulum produces the outer perivitellin layer, OPVL. The egg contains two chalazae, pronounced chalazi, singular ichalaza, which appear as white rope-like structures that project from opposite sides of the equatorial region of the yolk. A chalaza is composed of the same type of material as the OPVL, except that it is twisted. As the oocyte rotates in the shell gland, the chalazae are wrapped in albumin and become visible. The chalazae function to maintain the blastodisc, blastoderm, of the yolk in an upright position. There are two types of albumin, egg white, surrounding the yolk, an inner thick albumin that adheres to the OPVL, and an outer more fluid, thin albumin that surrounds the thick albumin. Thin albumin is surrounded by two fibrous shell membranes, an inner and outer shell membrane, which are separated by the air space located at the blunt end of the egg. The HDL gland, uterus, adds the calcareous egg shell. The porous egg shell lies next to the outer shell membrane. The egg shell is composed of three layers: an outer cuticle, an intermediate spongy layer, and an inner, mammillary, layer. The inner layer is composed of conical columns of calcite, calcium carbonate, which are deposited around the outer shell membrane with their conical ends closest to the shell membrane. Pores are present between the calcite columns. The spongy layer also consists of numerous pores allowing for gas exchange. The spongy layer is pigmented in colored eggs. The cuticle is present as the outermost layer of the domestic chicken egg, but is not present in all bird species. Mammalian cleavage. Mammalian embryos undergoing cleavage stages are referred to as pre-implantation embryos because they undergo cleavage before they undergo implantation, attachment to the endometrium, which is the uterine lining. There are 3 taxonomic groups of mammals, each of which exhibits a different cleavage pattern. The subclass Prototheria, monotrams, which includes the duckbill platypus and spiny anteater, has tchlolocythal oocytes and exhibits merolytic discoidal cleavage that occurs in the oviduct. The subclass, Theria, has 2 groups: 1, the Mctatherians, marsupials, that include the kangaroo, koala, wallaby, and opossum, and 2, the Cuthcrians, placental mammals. Most Metatheria produce mxolocythal oocytes, and exhibit holoblastic cleavage. Because fertilized oocytes are rapidly transported through the oviduct, Metatherian embryos cleave in the uterus, except the bush-tailed possum. The most studied eutherian embryo is the laboratory mouse that exhibits many similarities to human embryos, tables A10, 6.2. In eutherian mammals, mice, pigs, with isolecithal oocytes, cleavage is rotational, holoblastic, asynchronous, and relatively slow, occurring every 12 to 24 hours, Fig 6.36, Table 6.2, A10, A11. Following. Third, cleavage. The 8-cell mammalian embryo undergoes compaction, in which individual blastomers flatten against one another and are no longer discernible. The individual blastomers undergo changes in the cytoskeleton and cell adhesion molecules, and become polarized. The embryo daco-impacts and divides, and then is referred to as a morula. During cavitation, the embryo's outer cells secrete fluid into the morula to form a space called the blastococle. This occurs during compaction at the 8-cell stage in mice. When the blastocele forms, the embryo is called a blastocyst instead of a blastula. 4 days after fertilization, the blastocyst stage is present in mice. The blastocyst has 2 distinct cell populations: the inner cell mass, which is a cluster of inner cells that give rise to the adult organism and contribute to the amnion, and the trophylity, trophectode cream, which is the outer layer of cells that gives rise to the embryonic portion of the placenta, extraembryonic membrane. In eutherian mammals, cleavage occurs in the oviduct. Cilia of the oviduct propel the embryo to the uterus. Upon reaching the uterus, the embryo hatches from the zona pellucida, Fig 6.37. The blastocyst expands and is now ready to interact directly with the endometrium. Embryos at this stage are often referred to as pre-implantation embryos, since they will soon undergo implantation. During the blastocyst stage, the inner cell mass, also called embryoblast or germ disk, develops 2 germ layers, an upper pilast and a lower hypoblast. The process by which the 2 layers forms in the blastocyst differs between the 3 mammalian groups. The mammalian blastocyst at this stage is referred to as a bilaminar embryo, resembling the avian embryo in this regard. Cleavage in humans. Cleavage of fertilized human isolecithal oocytes is rotational, hololytic, and asynchronous. Human embryos cleave once a day until the 8-cell stage, when they undergo compaction to form a morula, Table 6.2, Fig 6.38. The blastomers divide more rapidly after compaction. A blastocyst, 32 or more cells, forms by 4 to 5 days after fertilization, Fig 639. After hatching on day 5, the blastocystic cavity, blastocele, greatly expands, Fig 6.40. By day 5, the blastocyst is ready to implant in the endometrium. The side with the inner cell mass is closest to the endometrium during implantation.